our next session is around eye-to-eye research and some opportunities. So we're coming back down south and we're in London at Great Ormond Street at Gosh. Is it Jonathan or Johnny?
You can call me Johnny.
Johnny, okay.
Yeah.
I'll let you introduce yourself, Johnny. Thank you.
Okay, sure. Thank you. I'm just going to share my screen. Okay. So unfortunately, Maria, my supervisor, Maria Musaji, is in Prague this weekend. She's developing some new gene therapies with some collaborators, so she apologizes for her absence. But I'm very privileged to be able to be here to talk to you all about the work that we did into Alström syndrome. So I'll just introduce myself. My name is Johnny Eintracht. I'm a PhD student, so coming to the end of my PhD, also writing off my thesis like Eleanor. And I was in the group of Maria Musaji at the UCL Institute of Ophthalmology. And I'm going to talk a little bit about a project that we did also in collaboration with Dr. Helen May Simmerer from the University of Mainz in Germany. So as part of my PhD, we were looking at developing a potential treatment for a subset of patients with specific genetic mutations that cause Alström syndrome. So I'd like to first give a bit of a background into our study and the rationale, and then I'll go through some of the science and the exciting results that we found. So Alström syndrome it's classified as a ciliopathy, meaning it's a disorder of the cilia. So the cilia are like the phone signal towers or receptors, the phone signal reception towers of the cell. And you can see a nice little image here from an electron microscope, seeing them on the lung, looking a little bit like a sea anemone. And they send and receive signals between the cell and its outside environment. But without these signals, so many important messages that the cells use to communicate with each other or to communicate with the environment would be lost. So without having cilia, it's kind of like having bad signal in your body, and the messages that need to be communicated cannot be communicated, and that can cause a lot of disorder. That can cause changes to a lot of organs in the body. But we coming from the Institute of Ophthalmology, I'll talk a little bit about cilia in the eye. Even though cilia can be found in almost every cell of the body and come in very different forms. But cilia in the eye, there's light sensing cells at the back of your retina known as photoreceptors, and they're made out of two main compartments. You have the outer segment and the inner segment. And these cells sense the light, and when they sense the light, they send a message from the back of your eye, from the retina to your brain, and this helps your brain make sense of the world around you. And these two sections are connected by the cilia. So if your cilia do not work properly and cannot send the signals properly as they should between different cells of the environment, the signals from your eye to your brain cannot be transmitted, meaning that whatever you're seeing in your eye cannot be sent to your brain, and it's hard to make sense of the world around you. And this can be this cilia dysfunction, or the cilia that don't work properly can affect other organs in the body where cilia are present too, such as your ears, in the cochlea, lungs, and kidneys also, amongst others. But Alström syndrome, as we've heard today, is caused by a genetic mutation in the ALMS1 gene or ALMS1 gene. And this, because of this mutation, cells can't produce a functional ALMS1 protein that the cilia need to work properly. So meaning again that the cilia, just to reinforce the message, the cilia without this ALMS1 protein, they can't send and receive signals as they should for the body to work, or to have good signal, as you can say. ALMS1, it's also so Alström syndrome it's a genetic condition, and it's a recessive condition, meaning you need 2 copies of the mutation from both parents to be affected. So if you have one, you'll be considered a genetic carrier, and if you inherit both copies, like child number 4 in this schematic, you'll be born with Alström syndrome. But But currently, as has been mentioned before, we don't really know what the role of the ALMS1 protein is still in the cell, and as such, there's also no current treatment available. So there's a lot of research still to be done, and something that we undertook to look at trying to find a treatment. Before going further, just a little background about what a genetic mutation is. There are 3 billion letters in your genetic code or your DNA, and a spelling mistake in this code is known as a genetic mutation. But if you imagine asking someone to type out 3 billion letters, most likely they're going to make a mistake or they'll introduce some typos, and our cells aren't really any different to this. And that means that all of us have have thousands of spelling mistakes in our genes, and a lot of them are harmless and don't mean anything. But when we have a spelling mistake in the ALMS1 gene, that can cause Alström syndrome, and these genetic mutations can take a few different forms. We focus on a specific form known as a nonsense mutation. So in this little illustration here from one of Maria's videos, on the left is the DNA code having a C and a G in its code, and then with a spelling mistake, this can change to a T or a T and an A, which can act as a genetic mutation and change how a gene works or make cells unable to produce the protein that they need to. So the mutation that we focused on at the type is called a stop mutation, or the technical term is a nonsense mutation, because when this mutation comes up, the cell it technically isn't able to make anything. It's kind of making nonsense. Is the protein generally the protein that's produced by the cell, and cells make these proteins from the signals that the DNA sends out as its instructions. And this is done, the instructions that the DNA sends out is in the form of mRNA and codons, which is the technical term. And these are really signals that the machinery in your cells read to make the protein that you see comes out at the top here. And your cells, in these messages, you have stop signals and go signals. So when your machinery in the cell comes across a go signal, like a green light at a traffic light, it says go, we're going to make the protein, and it keeps reading and reading, and the signal passes through the cell or passes through the machinery. And when it comes across a stop mutation, it shuts down and stops making the protein. But when you have this stop or nonsense mutation in your DNA, in your genetic code or in your DNA, this means that a stop signal is introduced early. So instead of making the whole protein, your cell comes across the stop protein when it shouldn't, or early, and shuts down, and the protein that the cell needs to make isn't made anymore. So in the context of LMS1, if there's a stop mutation that's introduced earlier into the LMS1 gene, the machinery in the cell reads this, sees the red light, and says stop. We're not making the LMS1 protein anymore, and it shuts down, which means that the LMS1 protein isn't made. However, there are certain drugs that can read through these stop mutations, kind of like this hacker on the New York streets, kind of changing the stop signals to go signals. The drugs don't necessarily do that, but they can come across a stop signal and then tell the cell machinery to not stop down, but to keep going, to keep moving the traffic, and still make the protein. So cells that are treated with these drugs, they may not necessarily lose the ability to make a protein, even if they have one of the stop or nonsense mutations in their genes. And this is very relevant to Alström syndrome, and why we decided to study it, because approximately 40% of mutations in the LMS1 gene that are linked to Alström syndrome are these stop or nonsense mutations. So this suggests that patients with these mutations may not have any ALMS1 protein in their cilia because the cell shuts down and doesn't make it as it should. And just to illustrate this, if you can see my cursor on the right, this is a nice picture of a cell from another study. And this green kind of tower, almost like snake-like shape, is the cilia, or cilium in singular form. And then this red dot here is the ALMS1 protein that's detected. So you can see that it's attached to the cilia at the base, kind of like the base that connects the phone signal tower to the power grid. So it's possible that we can treat patients with these drugs and help them to make or remake this ALMS1 protein that's connected to the cilia. So we looked at two specific drugs, and Kerry spoke about repurposing drugs, and that was a very important principle for this study. So one of the first drug that we looked at is called ataluren, also known as PTC124. And the key thing about ataluren is that it already has been approved for use to treat other conditions like Duchenne muscular dystrophy, that's also caused by stop mutations in a different gene. And it's gone through all the health checks. It's been approved for use, very safe, and can even be taken by young children. Ataluren is taken 3 times a day orally, and it's in a powdered form, so you can pour it into your orange juice or water or milk, or whatever, or your coffee in the morning, and then drink it and take the drug like that. We also looked at amlexanox, which traditionally for the last 50 years has been used for ulcers and treating asthma. It also has anti-inflammatory properties. So this was really a repurposed drug after intensive studies that were looking at a lot of different drugs, found that amlexanox specifically had this ability to read through the stop signal, or like a red light that a cell may have, or that in the instructions that a cell has to make a protein. So knowing that it could read through the stop signal and keep a cell making the protein, we decided that it would also be a great drug, or an appropriate drug to look at in this study. But in order to test out these drugs, we needed some patient cell models in the lab, and this led us to the patients. So the methodology here is to take a small sample of, it's like a whole bunch of skin from the upper arm, and we put it in the dish with some nutrients. And over a few days, the cells grow and proliferate and kind of take over the whole dish. And over time, they become nice looking skin cells. So you can see in this slide over here, this top right image this is a real life picture that I had from the work that I did of skin cells from an ALMS1 patient. And these are grown in dishes or flasks, like in this picture here. And the cells, we feed them every day with nutrients, or like a nutrient mix to keep them alive and happy. And when we want to treat the cells with the drugs, we can put the drugs into this nutrient mix. And just as the cells take up the nutrients that they need to survive, they also take up these drugs. This is a great way, or this is a very important way for the drugs to get into, to be taken up by the cells and to work on the cell machinery to reproduce the ALMS1 protein. Now getting on to the project that we did, we had 4 main research questions to guide us in this project. We first wanted to see whether we could actually detect the ALMS1 protein in the skin cells that we were growing up. And if we could detect the ALMS1 protein, we could also detect when it wasn't produced, if we were looking at cells in patients, in Alström syndrome patients. And then if we could show that it's detected, or we could detect this protein in cells taken from donors without Alström syndrome, and show that it's lost in cells from Alström syndrome patients. We can also show that it can be remade in Alström syndrome patient cells after treating them with the drugs. So that was our first question. The second question that we wanted to ask was whether the cilia actually looked any differently in the Alström syndrome patient cells. Not only whether they look any differently, but whether they act or function in a different way. And the fourth question, with the assumption that there are changes to how the cilia functions, can we restore function or rescue how cilia work after treating them with the drugs ataluren and amlexanox that can read through the stop signal that's introduced earlier into the ALMS1 gene, making the cells not produce the ALMS1 protein as they should. So just like the previous slide that I showed, we also we found ALMS1 as this nice red dot here. So this is actually the ALMS1 protein in cells that were donated from people without Alström syndrome. But you can see here, it's quite striking that in cells that were donated by Alström syndrome patients, we couldn't detect any presence of the ALMS1 protein. However, what was a very interesting and exciting result was that once we treated the cells with both Ataluren or PTC124 and amlexinox, once again we could detect the expression of this ALMS1 protein at the base of the cilia, suggesting that one, we can detect ALMS1 in the patient in skin cells. We can show that it's lost in ALMS1 patient cells, but then it's also restored or rescued in cells that we've treated with Ataluren and amlexinox, these read-through drugs. We then looked at as many cilia as we could in the lab, and we measured their lengths and we counted the cells that actually had cilia, but we didn't really see any differences, similar to a paper that Rob Semple published a few years ago, in the length of the cilia and the amount of cells that actually had cilia. And this suggests to us that ALMS1 doesn't really affect how the cilia are made, but it left the question open that perhaps the loss of ALMS1 affects how the cilia function. So we looked at a few things to answer this question, a few proteins. We first studied a transporter protein known as IFT88, and this protein carries the signals within the cilia. And you can see here, it's a nice green dot in our control cells. And it's also, there's another smaller dot that you can see fits into the cilium here. But again, remarkably, this machinery was lost. We couldn't detect it in cells from patients with Alström syndrome. But again, remarkably, once you treated these patient cells with Ataluren and amlexinox, we again detected the presence of IFT88, this transporter protein, suggesting that there's specific machinery or transport machinery in the cilium that are lost due to the ALMS1 mutation, but that once the ALMS1 protein is remade in cells treated with the drugs, we can recover the use, or we can recover this machinery again to help the cilium function. So not only did we look at the actual machinery, which kind of compares to a conveyor belt, as how I think about it, but we also looked at the signals of what the conveyor belts carry. So to do this, we looked at a certain protein receptor known as SSTR3, which is part of a lot of signaling pathways that the cilia use to communicate. And again, similar to how ALMS1 was, it's in a similar location or spot to where we found ALMS1 next to the cilium, we found SSTR3 connected to the base. But in the ALMS1 patient cells, we found that SSTR3 wasn't exactly at the base of the cilium, but it sat to the side, like you can see in this figure here, this diagram here. Which in my mind, I kind of thought about it as lost luggage that hasn't got to its destination. So it's been, you might have gone on a plane to the Caribbean and your luggage has ended up in Australia. And the cargo, what's needed to be transported just hasn't got to the right place. But again, once we treated our cells, or once we treated the patient's cells with Ataluren and amlexinox, we were able to show, or we saw that SSTR3, this protein cargo, kind of like the luggage of the cilia, again got to its right destination under the base of the cilia, as you see with this red dot here. So just to sum up everything quickly, there isn't an existing treatment for Alström syndrome, and certain stop mutations in the ALMS1 gene make the cell unable to produce the ALMS1 protein, and this affects cilia function. The treatment of ALMS1 patients or Alström syndrome patient cells with both Ataluren and amlexinox was able to rescue the lost ALMS1 protein and help the cilia function again. So it's kind of like having changing between good signaling in the cells to lost signaling in the cells, and then restoring that again after the drugs. We were also very fortunate to be able to publish all our work from this study in the eBiomedicine journal, which is part of the Lancet, one of the largest medical journals in the world, which was very exciting for us. And we can send copies of this paper out if anyone wants. Also, we have summaries for those without a scientific background, and that's if we can send that out through ASUK. You could say after all of this work, like, Johnny, this is great, but what does this really mean, or what comes next? So a few ideas that we had as a group was to expand this study to look at more patients and different stop mutations to show that these drugs are effective on as many patients as we can treat. We also, in this study is limited because we only investigated skin cells. We know that Alström syndrome as a syndrome affects multiple tissues or organs in the body. So it would be important to study more complex models of the disease, such as the retinas in the dish, which is what our lab specializes in making. So to grow up, and this is what I show in this picture here. These are some, they're known as retinal organoids. So these are cells that are made into like early retinas or even late stage retinas, and we grow them in the lab for up to a year, and they can really recapture what a retina looks like. So it would be an important future direction to be able to grow up these retinas in the dish from Alström syndrome patient cells and treat them with Ataluren and Amlexanox and potentially other read-through drugs to see if the cilia, if we can rescue how cilia function in the photoreceptors in the light sensing cells in the retina here. And again, like Eleanor mentioned, some animal model work would be very important to show that in biological systems these drugs are effective in treating the syndrome. And this would also push us to beginning phase 1 and 2 clinical trials after these steps or these future steps in the studies. And this is Maria. This is important for us as a lab group, as Maria Mustaji she's currently been given some Ataluren, which is one of the drugs that we use to investigate read-through in this study. She's been given Ataluren to treat choroideremia, which is a cell and eye disease, but also Usher syndrome. And Usher syndrome, it shares some clinical features to Alström syndrome. So if we see in these trials that Ataluren is able to alleviate or rescue some of the losses of sight and vision, sight and hearing in Usher syndrome through oral delivery of the drug, this is a really important and great step in the right direction for treating Alström syndrome and progressing into clinical trials with repurposed drugs. Our lab also has created a website known as Gene Vision, which is designed to help all those involved in rare diseases. It's a website full of very clear and helpful information. You can search ALMS1 gene or Alström syndrome on this website, and it gives you a nice summary and also helps. It provides links for further research. And if you just go onto www.gene.vision and then search Alström syndrome in the toolbar, you'll be able to, in the search bar, you'll be able to find all the information on Alström syndrome there. One of the most important, maybe the most important slide for tonight is just to acknowledge everyone that's been involved in this study. This study it's not possible without a team of brilliant scientists from around the world. So I'd like to thank my team, and particularly my supervisor, Professor Maria Mustaji from UCL Institute of Ophthalmology. We also had some assistance from Great Ormond Street, from Dr. Elizabeth Forsyth and her PhD student. And again, this study wouldn't be possible without our collaborator, Dr. Helen May-Simera from Mainz. I also wanted to thank Dr. Rob Semple, who's on this meeting, on this call, for sending us some Alström syndrome patient cells that we could look at, and also Dr. Jin Hua Chen, who was part of his team. Important to say thank you to our funders, and also most important to say thank you to the patients and their families, without whom none of this research would be possible. And I'd just like to end off by saying, if there's any questions or any further things you want to talk about, you can get in touch with either myself or Maria. And these are just a few pictures of patients that have come to donate skin samples or to participate in, not only to donate cells, but to participate in studies that our lab has conducted. So one of the, there's many capacities to be involved in research as a patient. One of the ways is to donate cells, and these could be grown up and tested in the lab, just like I've showed in this talk. But there's also natural history studies that we're interested in in conducting, and these help track the progression of the condition. And when we can track the progression of Alström syndrome, this also means that we can make objectives or landmarks for clinical trials to show that that maybe vision has changed over time. But once treated with a certain drug, vision is restored to a certain point, just like it was at a previous point at an earlier stage of the disease. So whatever contribution you feel comfortable with as a patient or as a family to make to research, please be in touch with Maria, and we'd be very excited to have you come into her clinic at Moorfields Eye Hospital and be involved in future research. So thank you very much for your attention and for being here tonight, and it's really a privilege to be here to talk to you. Thank you.
Perfect. Thank you very much, Johnny. I know we've been all quite excited by some of the early stages of the research that you are involved in. We have just time for a couple of questions if you're happy to take some of those. We will, of course, upload the links to your website and the publication. We will upload to our website too and share that with all of our attendees and others who look at the website. So have we a couple of questions for Johnny? I can see there's a red flag on there. I don't know if Catherine or...
Yeah, so let's have a look. So we have one from Jack Chu, and he says, if it can solve the cilia function, does it mean not only for eye, but also other organs of whole body? Finally, AS can be clued by TRID drug, TRID drug. Not sure what that second part is. But I suppose his first part is really asking around if we can use this to solve the cilia function, can we also use it for other parts of the body?
Right. So I think that's one of the advantages of having oral administration of the drug. So if it's something like Ataluren, which is a powder that can be put into anything that you drink, that can be, if by drinking the drug the body will be able to transport it to all over the body, and the effectiveness, we don't really know the effectiveness of this yet, and more trials are awaiting on finding this out. But in principle, it should be a whole body treatment or systemic treatment to treat multiple organs that are affected.
Okay.
Johnny, can I just ask a question? So it's from a conversation I've had with a family member before conference. So obviously these are drugs that are already licensed, not in Alström, but they're licensed. So there's already a safety profile there with the particular drugs. Is there a possibility of using these, using compassionate use in Alström patients? I know you've talked about other conditions such as Usher. Do we need to wait until those trials have concluded before looking at the potential of Alström patients? I just want to manage expectations for people as well.
Right. I think based on our study, there's not enough to go straight into medicating patients. I think further work needs to be done both on more complex cellular models, also on animal models, before looking at going into any patient treatments. I don't think it's entirely dependent on other syndromes, or other trials, but I think data from those trials would definitely expedite the process in getting approval. Again, I'm not a medically trained professional, unfortunately. I just work in the lab. So Maria, she's a better person to ask these questions to.
And we might be able to direct. So let's just have a final question. Catherine or Kerry, either of you? I know there's one question from Gonzalo.
Okay. I'll have another then. Is there any more questions, Catherine? No. Okay. So therapy, Johnny. I know obviously that's something that interests Alström families. Our concern, we hear time and time again, is the size of the Alström protein, and I know that there are studies going on, particularly in Usher, on non-viral gene therapies, and just wondered from your work in the lab whether technologies are moving on where the size of the Alström gene might not prevent gene therapy treatments in future.
It's a great question, and it's a problem that we encountered in our project, just in trying to determine the right technique to detect the Ulstrom protein, because it is so large. I think especially one of the reasons that Maria isn't here is that she's with collaborators trying to develop these non-viral vectors for gene therapies. These non-viral vectors, it's not necessarily my area of expertise, so I don't know that much about it, but I know that one of the improvements on traditional gene therapy is that their capacity is increased, so they're able to hold larger genes. So I think the results from Usher syndrome trials with these vectors is important, because the USH2A protein is also a very large protein, or it's a very large gene. And I think Ulstrom, the ALMS1 gene, is even slightly larger than that. So if that's shown to be transmitted to a cell, and the cell can take that up and then transcribe that gene, then that's also very encouraging for the use of the ALMS1 gene in gene therapy. So there are advances. Nothing has been shown yet, but there are advances, and it's definitely a question or an objective on a lot of researchers or lab groups' minds.
Great, lovely. It'd be great to follow up after then, if that's okay.
Yeah.
Okay, and there's a couple of other questions on there which I don't think Johnny can answer, but if we can capture those and follow up with one of the professionals afterwards, I think that'd be really helpful. Yeah, one is to do with getting the drugs in India, and one is to do with growing livers. So if we can capture those. Thank you, Johnny. Really appreciated that. I particularly liked your visual presentation of it. That helped really fix it in my head. So
Thank
really
you.
appreciate it. And we will put the publication, the links to it, and the links to your website on the on our website too. Thank you. Okay.
Thank you.